Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I'll be speaking with Benedict Cross, Chief Technology Officer at PhoreMost, for a conversation on the boom in targeted protein degradation, or TPD, market growth, and, in particular, how AI and computational small molecule drug discovery approaches are helping to realize this potential in molecular glue development. Welcome, Benedict.
Thanks for having me.
Now, PhoreMost specialises in next-generation TPD and is progressing a pipeline of degrader therapeutics within oncology and inflammation. But before we sink our teeth into our topic today, Benedict, or if I may refer to you as Ben, perhaps you could introduce yourself to listeners and share with us the passions and the journey that took you to doing what you do today at PhoreMost.
I'll be very happy to. Thank you. Of course, Ben is fine. Thanks, Nicole. So, my name is Ben. I'm the Chief Technology Officer at a company called PhoreMost. We're a UK-based biotechnology company based just south of Cambridge. And yes, as you noted, we've got a strong interest in the field of targeted protein degradation and progressing small molecule drug assets through that particular modality. As for myself, my background in research is in the field, what we call these days proteostasis. That's a field that describes the understanding of how proteins within cells and bodies behave, how they're turned over, recycled, and also functionalized by the body, and also crucially, how that process can go wrong in different kinds of diseases. That converges on targeted protein degradation for obvious reasons, because that's a field which is very much about understanding not just how proteins can be recycled and turned over, but also how we can do this deliberately. I'm sure we'll come on to that technology in due course. More recently in my career, I spent a lot of time in the gene editing field. I'm a functional genomics specialist. I worked a lot in the field of CRISPR technologies for many, many years before coming to PhoreMost. And from the technical perspective, that's a field which also is very, very important for what we do at PhoreMost. We have a proprietary platform, as we call it in the biotechnology field, which allows us to do all sorts of different interesting things at very, very high throughput. And that's how we found our way, somewhat serendipitously, but also partially by design, into the field of targeted protein degradation. I've got very excited about the possibilities that we have there. That's enough of a bit of a background for me. Happy to tell you a bit more about PhoreMost or dive into some of those subjects if you like.
Thank you, Ben. As you say, it's exciting, the field that we're going to be discussing today. So on that TPD technology and focusing on the market growth within that, targeted protein degradation via the proteasomal and lysosomal pathways represents a novel tool to explore cellular pathways and a promising therapeutic approach, as Ben has been discussing. The concept of TPD was first proposed in only 1999, a veritably revolutionary approach in drug discovery that focuses on the selective removal of specific proteins associated with diseases, as Ben has been discussing. Unlike traditional small molecule inhibitors that merely block protein function, TPD harnesses the cell's own degradation machinery to eliminate unwanted proteins, including those previously considered undruggable. So, Ben, I was wondering if you could provide perhaps a general history of the development of TPD and indeed discuss for me, for listeners, the recent boom in this market.
I'd love to. Let me just quickly, you mentioned something really nice there that's really important, which is about why TPD is important and interesting. And you've highlighted that most drugs, across the last 100 years, been designed or found by accident, but ultimately function in the same way, which is that they inhibit enzymes or pathways in a very direct one-to-one fashion. And the whole concept of targeted protein degradation takes things to a much, much different, more exciting place, because as you note, it allows us as drug designers and drug discoverers to come up with molecules or therapies which are much, much more potent, much more powerful, because eliminating a protein that's misfunctioning or is important for the driving of a particular pathway in disease completely from cells and from bodies of patients is a very, very potent and powerful way to treat those kinds of diseases. And there are many, many diseases to which that's applicable. So the idea is absolutely revolutionary. I would say it's actually relatively old now as well. As 1999 isn't so long ago in biology understanding terms, but actually for us to progress where we as a field have got to, getting small molecules close to regulatory approval, maybe taking a little bit longer than we might have anticipated. And it might be worth trying to think about why that might be, because it highlights some of the challenges that the field is trying to overcome at the moment, and us in particular. So maybe first of all, I'll dive into a bit more of the detail about how it works and why it's interesting. So as I noted before, field of proteostasis, or in general, understanding how proteins function in bodies and cells, a big part of that is understanding how they are ultimately gotten rid of naturally by cells. It turns out cells do that all the time. They have to do that because proteins only last so long, enzymes are only functional for so long, or they might go a little bit wrong, misfold, or get mutated or anything else. So the cells in our bodies and in all bodies of mammals and other species as well have had to come up with ways to get rid of those proteins. And they do that by tagging them with a little small marker, a flag. It's actually a small protein called ubiquitin. And that triggers a process which ultimately results in this proteasomal, or you noted also other routes to get lysosomal degradation, but gets rid of those proteins completely from cells, or actually recycles them into smaller units, and they can be turned into new proteins thereafter. So the idea that was come up by those years ago by Craig Crews and Ray Deshays and others advanced it since was, what if we could do that deliberately? This is the targeted part of targeted protein degradation. What if we had a target or a protein that we wanted to get rid of? Maybe it's in cancer. You want to get rid of one of these famous oncogenes that are, very, very difficult, but very, very important in the drivers of the development of cancer, MYC or KRAS. What if we could use this process that the cell already has, the dustbin machinery, and get it to be tuned to our purposes as drug discoverers and trying to find new therapies? And the way they did that was to first of all understand how the proteins do it. Yes, they have these little flags that they attach to proteins, but the trigger for that is sometimes described as a degron. It's a short sequence within the proteins which marks that protein as an old or misfolded or mutant species. They call it a degron. And the very first molecules that Craig Crews and Ray Deshays built literally took that small, it's a peptide sequence. So if you understand proteins, they're strings of peptides. They took 3 or 4 of those peptide sequences and they attached them to something else. That created these first bifunctional molecules. That's bifunctional because they have 2 ends, one which sticks to the target you're trying to get rid of, and one which sticks to the machinery, or which is recognized by the cell's machinery to drive that degradation process. This was called PROTAC1. Now, of course, the field has advanced substantially from then. And as drug discoverers, the medicinal chemistry quest that has gone on has been nothing short of incredible. They've been able to, across many, many different companies and academics and hundreds of and thousands of different papers published, move from that PROTAC1 into many, many hundreds of variants of that, both which target different proteins for degradation across different diseases, from oncology and neurodegenerative diseases and beyond, but also using lots of different parts of the cell's machinery. There are hundreds of proteins or enzymes which are really useful in this process. And understanding how many of these can we recruit, can we hijack, can we co-opt to do this job for us has been a big part of it. And shrinking these molecules, which are otherwise quite big, down to more traditional, smaller molecules, literally in the number of atoms associated with them and the way they can behave predictably in patients' bodies, has been a massive challenge. And that explains why it's taken us over 20 years to get to the point where we're just on the cusp of regulatory approval by the FDA for the first most advanced PROTAC molecules. It's a very, very difficult job, but it's so exciting and so powerful that so many minds have been applied to it. And we're now starting to see the fruits of all that effort across lots of different companies and academic groups combined. You noticed a bit about the market boom. I think that's probably just one final sentiment around that. I hope I've explained the technology from the technical perspective in enough detail to understand that the promise of it is very vast. And it really does have this huge, broad potential. And in particular, over the past 10 years, that creeping forward with these bigger complex molecules as we started to realize that it really was achievable to have drugs which look like this, has been very exciting. But maybe over the last 3 or 4 years, that's sort of shifted again in a little bit of a sea change in the ideal properties or features or designs of these kind of molecules. And these days, more and more people are interested in trying to shrink those molecules down into much, much smaller, more traditional drug-like shapes. The reason to do that is because if you're a small molecule, fewer atoms, better predictability, easier to synthesize, but crucially also behave more like a drug that you can pop in a tablet and give to a patient without it having undue off-target or side effects, or just not having sufficient degree of efficacy, as we describe. And that quest, journey, and desire from scientists has led to a really big excitement in the field of molecular glues. We in particular at PhoreMost have been very motivated about that field, and for the past 3 years have been trying to understand it in much greater detail and come up with technologies that I'm sure we'll have time to discuss that can address this slightly different twist maybe on the targeted protein degradation field.
Thank you, Ben. So incredible indeed, as you've been describing, with the vast promise and potential of these technologies. So if we do turn now specifically to molecular glues and that different twist, I believe PhoreMost recently published a study demonstrating the capabilities of its high-throughput GlueSEEKER platform, which accelerates the design and development of novel molecular glue degraders. That paper, entitled Systematic Molecular Glue Drug Discovery with a High-Throughput Effector Protein Remodeling Platform, was published in bioRxiv and describes PhoreMost's unique approach to molecular glue discovery. So I was wondering, Ben, if you could tell us more about that approach and indeed more about the GlueSEEKER platform itself.
Yeah, fantastic. Thank you. Yeah, we were very happy to deposit this paper recently, share it with the rest of the field. We think this is a very exciting area. I'll tell you a little bit about that in a second, but I think it's probably worth me also dwelling a little bit on what is a molecular glue. Lots of details here, but I hope you don't mind me diving in a little bit where I get a bit too excited. But
we like details. Go for it, Ben.
Okay, good stuff. So targeted protein degradation, for the past 20 years has been predominantly associated, or at least much of the work and the kind of the themes developing have been about these bifunctional molecules, or PROTACs, which stands for proteolysis targeting chimeras, which were invented by Craig Crews and Ray Deshaies, as I mentioned. Actually, in many ways, the molecular glues world is much older, but only recently have we really started to squish them together and start trying to understand how they compare and contrast and what's the most desirable or most appropriate different kinds. The difference is a little bit subtle, but let me explain them. The outcome is the same. The desire of both a PROTAC or a scientist designing a PROTAC and scientist designing a molecular glue is the same. You're trying to stick two proteins together or bring two proteins into proximity and get a functional outcome, one protein killing the other one or one protein doing something to the other one. Ultimately, that's the aim. Molecular glues are, by definition, much more traditional, small, fewer numbers of atoms, molecules, and that has all those benefits of that I've touched on previously from the medicinal chemistry perspective. The difference in the way they work is that they tend to have lower sticking power, lower affinity, and they tend to slot into smaller gaps, as you can imagine, between these two proteins. But the biggest difference is that is a really fundamental discovery difference that nobody knows how to discover them. They've all been found completely by accident or by building on accidental discoveries. The most famous molecular glue in terms of medicine development were based on the thalidomide analogs. They're, of course, famous for all sorts of terrible reasons. But late on in the understanding scientifically of how those molecules work, long after they were withdrawn for the treatments that caused teratogenicity in pregnant patients, it was discovered that those molecules actually function as molecular glues and they stick two proteins together. Crucially, one of those proteins is a member of this degradation pathway, an E3 ligase, so it's able to degrade other things. And now we even start to understand how that can cause these horrible teratogenicities. But it also allows scientists to think about what else can those molecules do. And because they're small and they are predictable and they're easy to get through the body and get to diseased tissues, get to tumors if you're treating cancer, and do a really impressive job, they're in many ways more attractive for drug designers. But the big challenge has been robustly, routinely, systematically, whatever phrase you like, again and again, deliberately finding these molecules, because the design principles that kind of define them are somewhat nebulous, by which I mean they all look a little bit different. The surfaces that they could stick to on proteins could be very shallow, not sort of traditional big deep pockets on enzymes like kinase inhibitors would bind to. They have massive potential, even bigger than PROTACs, because the properties that define their features are even less sort of discrete. In some ways you can, as I said, they can be less sticky, lower affinity. They can have all sorts of different freedoms associated with them. But for many years, us and others have been trying to come up with routine ways to find these things. Before we came into the field, how was that done? That was done by guesswork and throwing lots and lots of molecules at problems and seeing if you get lucky. That's broadly speaking how the pharmaceutical industries have approached it. It's cost a lot of money. A couple of molecules have come out of it, but it has not been a cost-effective process. So it's been very important and a big recognition in the field, especially in innovative biotech companies, see if we can come up with new ways to do that. Our approach is this GlueSEEKER technology that we deposited recently, and has been really, really well received. And it sort of fits into an emerging wave of ideas. And the ideas, broadly speaking, are about if you could understand general principles of how two proteins might come together in the absence of the molecules, could you actually then start to think about how you could make a molecule that could do that more frequently, or to induce that event, two proteins coming together? Because two proteins sticking together is not quite enough. You need them to stick together and then stay together just for a little bit of time. And that a little bit of time allows the interaction to generate a functional consequence, a tagging with these ubiquitin molecules or something else if you're thinking in another field. So there is a requirement, a little hump to get over, sticking two proteins together and then having them stay together long enough. Our technology allows us to deliberately and prospectively screen protein surfaces for modifications, mutations, loops, insertions on those protein surfaces, which does exactly that, sticks that protein to something else. So generally speaking, we've been doing this on the E3 ligase side, or what we sometimes term the effector protein side. So the effector protein is the kind of active part of the equation of those two proteins coming together. And so we take an E3 ligase, such as cereblon or other ligases we've highlighted as well, and we systematically edit that from a protein perspective. So introducing subtle changes to the surface topology and subsequently the chemistry of that particular protein. And we ask a very direct phenotypic question. We say, okay, well, now we've made hundreds of thousands of minor modifications to that protein sequence. Does it now do something new? Because broadly speaking, these proteins work by interactions. That's how they function. And we can screen for that very deliberately because, okay, does it now, in fact, even degrade a highly important, highly validated oncology target? Or does it now degrade mutant tau or Huntington in these different neurodegenerative diseases? And we can screen for that very deliberately in cells, which is crucial because it gives us a really precise, relevant physiological readout. And when we found those things, which it turns out we could, we've then been able to do the kind of magic bit afterwards. We say, okay, now we can edit this protein. So it's a slightly different sequence. And I'm talking just about putting 2 or 3 or 4 amino acids in there that weren't there before. Find that it now sticks to something and degrades it. And we can now go back in and reverse engineer that loop into a small molecule. Because really, just as if we harken back to the first bit of our conversation, the first PROTACs were based on peptide sequences, they're based on degrons. And in the past 25 years, not just in the protein field, but all across biology and drug discovery, it's been more and more development and understanding of how do the basic fundamentals of the chemistry of proteins sticking together relate to small molecules. What are the interactions, the hydrogen bonds, the electrostatic considerations, the geometry of molecules that need to stick into these little tiny holes between two proteins? And how does that peptidic sequence from the protein tell us how to make those molecules? And that's what we've been doing. And maybe this is a moment to then come to something else which I think is really important, which is the computational part of this. We sit in a world in biology and drug discovery where AI is increasingly pervasive and important and super, super helpful. I think this whole field of molecular glues with these little inserted sequences I described from our platform is a beautiful, perfect example of how you can enable computational drug discovery. Because the biggest challenge with computational drug discovery is the data in part, where AI-based molecule improvements or discoveries have been made most effective has been on paradigms where there's a lot of data, lots of similar molecules, lots of similar proteins, lots of real crystallography. And as soon as computational approaches take a leap into maybe the unknown, or where there's not enough deposited data, or we're starting to guess too much, everything comes crashing down. Now, our technology provides really, really rich data input into computational drug discovery approaches. And that's exactly how we found our molecules. We did it all in silico. We found molecules which looked, the computational modeling, that they'd slot in there very nicely. And when we take those through into the lab, we find that we get an astronomical increase in the number of successful hits from that. So we move from anticipated 0.1% hit success rate in any general sort of discovery campaign to maybe 5, 6, 7, or even 10% success rate for the molecule we're finding. So that is a huge leap in success, gives our chemistry teams far, far more to work on, and obviously is a huge saving in time and effort in the whole drug discovery process. So there's a really neat coming together, a convergence of these two different areas of targeted protein degradation and molecular glues and AI-based drug discovery. GlueSEEKER sits right in the middle as this enabling technology, which is super exciting for us.
Very exciting indeed. And as you say, it's all the puzzle pieces coming together after many years of research and hard work. So, my final question for you today, Ben, is of course pertaining to the future. What do you see the future horizon of TPD, of molecular glues, looking like? Is it to be an upward trajectory from here, or will there be further challenges, in your opinion?
It's a great question. I'm very excited about the future, but I'm not unaware of the challenges ahead. Absolutely. The excitement comes from the kind of things we've discussed today. These technologies, which are, all big leaps in drug discovery, come from a synthesis of different areas. And this is a really, really good example where you're getting a lot of things coming together at the right time. I haven't even mentioned the sort of augmenting technologies, and there are many, from mass spectrometry to even next generation sequencing, which continues to become a barrier reducing approach to doing these things at high throughput. There are some big strides to still make in our understanding of medicinal chemistry. So yes, computational approaches can be very, very useful, but there are still many, many challenges when it comes to making an actual functional medicine out of these things. We can discover things in the labs and in vitro, and even as we do in cellular systems, much, much more routinely than we can actually translate them into new medicines for patients. And the hurdles associated with that are yet to really even be properly understood when it comes to molecular glues. I've described a very strong degree of optimism that because they're more kind of conventional molecules, those things should be a little bit more predictable, more traditional. But we certainly have seen from the PROSAC field that it's been anything but that. So we shouldn't be blind to the possibilities that these are very complex cellular systems that we are recruiting and hijacking, of course. Enzymes being repurposed in cells, therapeutic potentials are obviously real, but there may be some downsides, safety and tolerability and tox, and that needs to be properly understood in due course. So I think scientifically, there's some super exciting things to see. More technology development of the kind that we've described. Genomic tools be really, really important as well. And I can see that very much as an upward trajectory. Approvals and regulatory steps and translation is always hard. The one final piece of the puzzle, which wish we didn't have to talk about, but is real, is the changing landscape corporately and politically. The macroeconomics that affect our ability to do the things which can yield great and exciting things have changed a little bit in the last even year, really. And that can slow the pace of development down. In other times, it can also focus the minds of scientists, and we can not be too gloomy about the future, because I think there's so many things to do, and we have much better tools and suites of software and opportunities and technology that we didn't have previously. But we need to be mindful that we've got to be giving the best experiments the best chance to succeed. And doing that on a daily basis, as well as across all of the whole industry, is really, really important for everyone. So challenges ahead, of course, but I, generally speaking, am much more on the optimistic side for these fields to continue to blossom and deliver new medicines, hopefully, to patients in the coming years.
A glass half full mentality indeed. Thank you very much for your time today, Benedict. It's been a pleasure.
Thank you.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments, at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.